Hi, this is Tiffany Martin with Medical Microbiology. We are in Chapter 5 and we are working our way through cellular respiration. We have gone through glycolysis. The end products of glycolysis was pyruvic acid, and we actually had two pyruvic acids. Now, the next couple of things that have to occur are really just to get pyruvic acid ready to enter the Krebs cycle. If we're talking about cellular respiration, we can take the precursors, the pyruvic acids that were the end products of glycolysis, make them the precursors for this next pathway, do a couple of things to create acetyl-CoA. And acetyl-CoA is what's going to be able to enter the third stage of cellular respiration, which is the Krebs cycle. So what things need to occur? Well, there's not a lot of consensus in what we call this stage, and so I kind of refer to it as the prep steps. You'll hear intermediate steps. You'll hear acetyl-CoA, synthesis of acetyl-CoA. You'll hear pyruvate oxidation. So there's a lot of different names for this, but the steps are the same. So let's look at the steps. All right, so we are going to start with pyruvic acid, and that's what we got from glycolysis. Now, cellular respiration is a very good way of making ATP. That chemiosmosis is going to generate a lot of ATP, so it's very efficient. Aerobic respiration using oxygen is the most utilized way of making ATP by all organisms on this planet. All animals undergo aerobic cellular respiration. All plants, all fungi, all protists, most bacteria. Now, there are a few bacteria that don't, and we'll talk about those few exceptions, but this is the main process, and it's almost virtually identical in animals and us as it is in bacteria. So let's continue learning this. So we're going to take a C3H4O3, which is our pyruvic acids from glycolysis, and let's see what happens next. All right, I'm going to switch to my board. There's my pyruvic acids. Remember, I got 2 pyruvic acids per molecule of glucose. So I'm going to put a big 2 in front of everything we do. So keep that in mind. All right, first thing that happens is to pyruvic acid is it is going to get decarboxylated. We are going to remove a CO2, which is called decarboxylation. So first thing that happens is we remove CO2. So I started with C3H4O3. I'm going to remove a carbon and 2 oxygens. I end up with an intermediate that is C2H4O from that. You can see how this is being removed. So that's the first thing that happens, decarboxylation. Now, next thing that happens is we're going to get some more oxidation and reduction. Remember, all of this is oxidizing glucose to make ATP. So there's going to be some more oxidation and reduction, some redox reactions that come in. So NAD plus is going to come in and get reduced to form what? Are you writing it too? It'll help you if you write it too. NAD plus is going to pick up some hydrogens and become NADH from that. Ooh, where do you think that NADH is going to take those hydrogens? You know it, don't you? To the electron transport chain. That's where we need a whole bunch of hydrogens. So every time we make an NADH, we know where it's going and we know why we need it. Because if we don't make ATP, we die, right? Okay, so that happens during these prep steps, these intermediate steps. We have now made some acetyl groups. We've removed some hydrogens, so we've made some acetyl groups called C2H2O. So we have some acetyl groups that have been made. We need one more thing to happen. We need something called coenzyme A to come in and make acetyl-CoA. Sorry, there's a lot of writing without you guys. So my acetyl groups were made after we removed the hydrogens, and now coenzyme A is a molecule that comes in, joins with these acetyl groups to form acetyl-CoA. Acetyl-CoA is what's going into the Krebs cycle, which is the next stage. So these are just kind of the steps that have to occur to get into the Krebs cycle. Now remember, we're keeping track per molecule of glucose. So I had 2 pyruvic acids. So how many carbon dioxides got made? 2. How many NAD pluses were needed to make NADHs? 2. We made 2 NADH. We need 2 coenzyme A's to make these 2 acetyl-CoAs. So don't forget to put the big 2 in front of everything because we're keeping track per molecule of glucose for that. Okay, so this are the prep steps, the intermediate steps. We see what has to happen, and we are ready for the Krebs cycle. Didn't make any ATP here, but that's okay. We did make carbon dioxide. Were we trying to make carbon dioxide? So carbon dioxide is considered a byproduct of this reaction. You can see where it shows up here. All right, so I'll catch you in the next video and we'll do the Krebs cycle. Thanks.